vesicular fusion and release of contents. So what does a synaptic vesicle then really look like? It's not these idealized cartoons that we tend to draw. It is a lipid bilayer that is absolutely chock-a-box full of proteins that do all sorts of things, right? One of those is this protein synaptobrevin, and it's the focus of the botulinum toxin. These proteins are going to do a number of different things, but in the case of vesicular fusion, the SNARE proteins, of which synaptobrevin is one of, are these proteins also known as SNAP receptors. And these are the proteins that are essentially labeling both the vesicle. The vesicle has certain vSNAREs, vesicular snares or vSNAREs, that give it its specific character, right? vSNAREs are going to bind to very specific T-snares, or you can think about it as target membrane snares or target snares. The vSNARE and the T-snare does two things. One is it helps to localize the vesicle to its proper location. So you can think about it almost like zip codes, right? My address is my T-snare, and the zip code on the vesicle is the vSNARE, and only when they find each other will they induce a fusion event. Okay? So you have vesicles that are going all across the cell, and they need to fuse with certain domains or certain membranes and not others. Okay. So that's one, is that it helps target, right, the vesicle to the right membrane. The other is that when they bind each other, they undergo a conformational change that's coordinated with one another. So they look like this, they bind, and then they start wrapping around each other like a rope. So they form what we call a coil-coil structure, and it's just like a rope that you twist, right? If you hold one steady and you twist a rope and keep twisting that rope, all of a sudden it's going to start kinking and the length of that rope starts shortening, right? That's essentially what snares are going to do. All right? So synaptobrevin is the vSNARE that's found in synaptic vesicles containing the neurotransmitters. Here's synaptobrevin. It's going to bind syntaxin on the on the plasma membrane. It's going to undergo this sort of wrapping around one another, and as a consequence, it pulls those two membranes ever so close, squeezes out every single water molecule between the phospholipids, and then they can fuse. All right? What botulinum toxin does is it's an enzyme that's going to cut synaptobrevin. All right. So how botulinum toxin works, the bacteria invades the gut. It starts producing botulinum toxin. Botulinum toxin is going to multiply and secrete the toxin. The toxin is actually going to traverse the gut, the intestinal epithelium. And we haven't defined epithelium to this point, but there are mainly two types of cells in the body, what we call mesenchymal, or cells that are predominantly adhered to their extracellular environment, right? And epithelial cells, which are primarily using cell-cell contact for their adhesive mechanism. And what epithelial cells are going to do is they're going to form these large sheets. And essentially, their goal is to create barriers. Our skin is an epithelial barrier. Our eyes, our mouths, our lungs, our intestines, everything that faces out to the environment, that is an epithelial barrier or an epithelial sheet. So the intestine, yes, is exposed to the elements through my oral cavity, right? And so it is technically facing the outside. The toxin actually traverses, right now, one of the primary hypotheses for how it gets across that, which would normally be impermissive for large proteins like the botulinum toxin to get through, they're a process known as transmigration. That's not right. What's my brain? My brain is frying. I'll get it to you guys. It's, I hate it when my brain does that. It's a process that a lot of bacteria, a lot of viruses, but also our cells of our immune system have evolved this mechanism to basically migrate right through a cell. Okay? And it's some pretty interesting mechanisms. All right, gets into the bloodstream. It makes its way to the motor neurons, and it's internalized by that process that we've highlighted before, known as endocytosis, right? So endocytosis, the cell is going to form a vesicle on the plasma membrane. It's going to pull in proteins and products from its extracellular environment. It's going to pinch those off and bring them in to the cell, endocytosis, as opposed to exocytosis, where it's pushing them out into the extracellular environment. Okay. So if we look at that domain architecture of the toxin, and we look back at those three domains, what we find is that there's a receptor binding domain. And so that receptor binding domain is what's going to bind to the motor neuron, and that's going to actually trigger a phagocytic or an endocytic, in this case, process. So here it's triggering endocytosis. This domain, this translocation domain, remember we talked about how in endosomes they become more and more acidic, right? As they usually traverse towards the lysosome, they're going to become more acidic because they have proton pumps that are pumping protons into the lumen. That translocation domain is pretty clever. It actually will undergo a huge shape change under acidic conditions, and it acts like a harpoon and basically will embed itself in the membrane of that endosome and bust it, right? So it destabilizes that membrane, the membrane bursts, it expels its contents, and now there's a cleavage event that occurs between the catalytic domain and the rest of this molecule. That's where they figured out there's a light chain and a heavy chain. Okay, so the light chain is the catalytic domain. All right, so this is just me writing it out. Toxin escapes the vesicles through that harpoon mechanism. The light chain separates from the rest through a proteolytic cleavage. And the light chain is what we call a zinc endopeptidase. So this is in a large class of proteins known as metalloproteases. It means they need a metal ion. They need a divalent cation usually, in order, always actually, for their activity. Endopeptidase means it's going to cleave in the middle of a protein, as opposed to an exopeptidase, which chews from the edges, right? From the N or C terminus, an exopeptidase will start chewing up. Endopeptidase is cut right in the middle of the well, not right in the middle of the protein, but you get the point, in the middle of the chains. All right, so if we cleave synaptobrevin and there's no synaptobrevin, there's no vesicle fusion, no neurotransmitter release, and all of a sudden I can't contract my muscles, okay? There's paralysis. All right, so remember enzymes are catalysts. The real problem here is that the toxin keeps catalyzing the reaction, the same reaction over and over again, and it can survive for days. So the real question is, how much damage can it do? All right, so last problem. Let's test our understanding now of the concept of turnover. So I'm going to give you a couple facts. For botulinum toxin, the Vmax is 14 micromolar per minute at an enzyme concentration of 100 nanomolar, molecular weight of 50 kilodaltons. What is the turnover rate for botulinum toxin? Can anyone remind us from the pre-lecture what turnover rate is? You guys remember? Yeah. Yes. The number of reactions per second per catalytic domain. Okay? All right. So I'm going to give you, we've got a little bit of time, so I'm going to give you a minute or two to work through this problem. And then we'll see what our guesses are. is in reactions per second. So it's a per second. Megawatt? Molecular weight. I'm sorry, yeah, molecular weight is 50 kilodaltons. Kilodaltons. Okay, does anybody want to fathom a guess? Yeah. 2.8 per second? All right, how'd you get the number? Okay. All right. Anybody else? It's actually not far off. Anybody else want to take a stab at it? All right. Let's work through it. So remember that, again, the turnover is kcat, right? It's the maximum number of conversions of substrate to product, which is what our Vmax is telling us, right? Vmax is 14 micromolar per second. Okay? Oh, thank you. That would be important. I'll get way off. All right? But it's defined at a specific concentration, 100 nanomolar of enzyme. All right? Okay. So 14 micromolar per second. God bless America. Holy moly. Per minute. All right, we can convert that to seconds, right, if you want, which I apparently need to do. 1 minute over 60 seconds, right? And then times divided by, right? Divided by the concentration of our enzyme, right? So that's going to be 100 nanomolars is how many micromolars? Anyone know that off the top of their head? 10 times, right, 10 to the negative 3, right? So 1 nanomolar is 0.1 micromolar. All right, so micromolar cancels, minute cancels, all right? 14 divided by 0.1 is 140, right? You just move the decimal place over 1. Divided by 60 gives us about 2.33 product per second. Okay? At Vmax, that's how much product per minute we're making. We convert that to seconds per, so this is why I made a big point out about saying per single catalytic site. That's why we're dividing by the number of moles of enzyme. Because we're not trying to figure out how many per 100 nanomole of enzyme, but per one molecule, okay? All right. Just because we're at the very end, what else would you need to know in order to predict how damaging a single molecule of toxin would be? What else is missing from this paradigm? I'm telling you, this thing can catalyze, a single molecule catalyze 2.33 reactions per second. How fast will it kill a neuron, a motor neuron? What? Yeah. That's important, but for a single neuron, since we're out of time, I'll go ahead and tell you, you need to know the amount of substrate, right? So it turns out that there are about 70 synaptobrevins per vesicle, and there are about 150-ish vesicles per motor neuron. So you can do the math and figure out it takes actually around an hour when you factor in diffusion. In about a few hours, one single toxin can eliminate the activity of a single motor neuron. Okay? That's why it is so bad. The treatment are inhibitors, right? Antitoxins, antibodies that block the catalytic site. All right, so thus far in class, we've talked about membranes, we've talked about enzymes, and now we're going to talk about the cytoskeleton. And we're going to do this in the context of a disease known as listeriosis. Listeriosis is a pretty clever bacteria that's evolved a lot of interesting mechanisms to both invade cells, to evade our immune system, and it does it through hijacking our own regulatory mechanisms of the cytoskeleton. So this bacteria is going to wind up leveraging things that we've learned about in the membranes, things that we've learned about with enzymes, and now things that we're going to learn about cytoskeleton. So in the pre-lecture, Dr. Janes sort of began to define the parts and pieces of the cytoskeleton, the nuts and bolts. What we're going to do in this lecture is start talking a little bit about regulation. And in general, that's how the pre-lecture and lectures will go, is the pre-lecture is all about arming you with who are the main players, and then in the main lectures, we're trying to describe how they actually work. Okay? All right. So listeriosis, much like the last bacteria we talked about, infects the GI tract and can move to the central nervous system. So it's very similar in that regard. It's caused by a bacterium, Listeria monocytogenes, or Listeria. And it also is a soil bacteria. Okay? It's a bacterium that becomes what we call an invasive pathogen. And does anyone want to fathom a guess of what invasive pathogen might mean? Take a stab at the definition. Anyone? Yep. Oh, lifting your desk. Okay. All right. Yes. Yeah, invasive maybe means attacking, right? Or invading, as the name implies. And so these are very aggressive, attacking bacteria. Okay? In mammalian hosts, and they do this through the expression of what we call virulence factors, or virulence factors. What these are either small molecules, proteins, biomolecules that enable their invasive behavior. Okay? And so we'll talk about the virulence factor of Listeria at the end of this lecture. All right. Guys, I'm fighting a migraine, so just bear with me today, okay? It's not full on yet, but if I start rubbing my head, that's what's going on. I'm okay. I'm okay. I've taken some meds. I don't think I'm loopy yet. All right, so much like botulism, you get this by contracting or eating, contracting the bacteria by consuming improperly handled foods. So raw meat, unwashed vegetables, again, it's coming from the soil. So as long as you're not eating copious amounts of dirt and washing your veggies, you should be okay. The prevalence is, this is backwards. Yep. Anybody check that out? Anybody find that? Prevalence is what? How many people have it? Incidence is what? A probability statement. So the probability statement, we've inverted them. Oops. So incidence is about 0.3 infections per 100,000 people. It's right on your slide? Okay, I must have pulled. Yeah. And so the incidence is about 0.3 infections per 100,000 people, and the prevalence, so as long as it's right on your slides, is 1,600 infections and about 260 deaths annually in the United States. Anyone give any good examples of recent outbreaks of Listeria from the news? Who here eats Chipotle? I used to. Yeah. Well, they've cleared it up, but I think it was last year or the year and a half ago, Chipotle had an issue with Listeria. I really like ice cream, Jenny's ice cream, if anyone's familiar with that. Before I moved from Atlanta, they had an issue. And about 5 years ago, there was a huge Listeria outbreak through cantaloupe distributions. So, yeah. So watch the news. All right. So what are the symptoms and complications? So Listeria is of greatest concern for people that, in general, categorized by a weakened or poor immune system. And so that's the one thing that all these folks have in common. Older adults, pregnant women, newborns, and other immunocompromised individuals. And these are what we would call risk factors. So risk factors refers to sort of, I would say, characteristics of a population that, as the term implies, gives them an enhanced risk of contracting a specific disease. In this particular case, the risk factor is a compromised immune system, which would tend to be a risk factor for just about everything. Yeah. What's that? I mean, yeah, so that's the comment I just made. It seems like having a weak immune system is a risk factor for just about everything. But this in particular, if you have a fully functioning immune system, you do not, you can contract the bacteria, but you don't tend to suffer from the same critical issues that they do. Yeah. But you're absolutely correct. Having an immune system is pretty important. Go ahead. Yeah, so virulence factors are the molecules, biomolecules, either proteins, lipids, carbohydrates, that are enabling the pathogen to become invasive, right? So it's what's enabling. It's almost, you could almost think about it as the protein that enables a mechanism of action. Okay? All right. So the symptoms, as you can imagine, then are host dependent. If you're healthy, then you get what's known as a non-invasive infection. You'll get it in the gut, and your immune system pretty much handles it there. These are quote-unquote mild effects, mild symptoms, diarrhea and fever. The fever is a natural host response. That's our natural response to pathogens. We raise the temperature of our bodies, and that begins to inhibit the ability of pathogens to continue. So it's a kind of a normal defense mechanism. In pregnant women, they'll suffer from fever, muscle ache, diarrhea, the same types of things, but this can lead to miscarriage, premature delivery, and infection of the fetus, which would be very bad. Other high-risk people, again, suffer from many of the same issues, but also confusion, loss of balance, convulsions, and most importantly, meningitis, which can ultimately kill you. All right, so Listeria invades the GI epithelium. Anyone want to remind us what an epithelium is, as I've defined it? Yep. Yeah, so it's a cell barrier. So just be careful about the scaling, right? We're going to learn how to be very precise. So yes, they have a membrane that faces the outside, but when we talk about an epithelium, we're talking about the actual sheet of cells, not just their membrane, but the actual cells themselves. And they're forming a sheet, remember, by associating with one another through what we call cell-cell adhesion and forming these really tight junctions that we'll talk about in the future, right, that prevent anything from moving in between the two cells, right? Okay. All right. So Listeria allows cell-to-cell transmission once they become invasive. So once they get out of your gut and into your system, they undergo this basic life cycle where it will, similar to the other, the toxin that we talked about, botulism toxin, this has cell surface proteins on the coat of the bacteria that activate phagocytic or endocytic mechanisms of your cells. So the cells will actively endocytose these bacteria. Again, they will also evade. They have mechanisms to evade those endosomes. They burst the endosomes and they get out into the cell body, into the cytoplasm. Here, what they'll do is they express a protein, Act A. We'll go over what that protein does, but that is their virulence factor. This is what makes them so invasive. Okay? Where's my mouse? There it is. Okay. All right. They'll divide within the cell, right? So there's division. And this is not just a cartoon description. These things are actually called comet tails. And you'll see them in a video here in a minute. These things form what's called comet tails that propel them forward. And so these are projectile bodies. They can, in fact, use that projection, that projectile motion, to actually impact your cell membrane and pull all the way out of a cell. And now they're completely enclosed within a membrane. And so in this way, they can begin to evade your immune system. Yes?
Do we have mess up the cytoskeleton so the cells can barge right through it?
Yeah, pretty bad, right? The cell can actually, pretty bad for cells to be punching holes in them, just in general. A lot of cells repair their membranes pretty quickly, but still not a great thing to have something rifling through your cells, punching holes in them. What it does is it can actually use that membrane, as you see here, to become stealth to the extracellular environment, right? So immune cells that might be monitoring this area, and use that then to invade neighboring cells, right? So once they get into the neighboring cells, again, they use those similar mechanisms to evade and get out of these early endosomes and endosome vesicles, and then continue their life cycle by dividing, hijacking this cell's cytoskeletal machinery, and moving into the next cell. So here's a pretty cool video that shows you what they look like inside the cell. So these little things that you see on the back side that almost look like shadows or little comet tails, that is that cell's cytoskeletal network working really hard for that bacteria. My crazy anecdote about Listeria is that when I was at Georgia Tech, there was a professor that decided it was a great idea to use Listeria as a drug delivery vehicle. And so he actually began to try to engineer Listeria to deliver therapeutic genes, but taking advantage of their ability, their virulence, right, their invasive behaviors. I don't think the project ever quite got off the ground. No one's putting Listeria in me. All right. So let's talk now about the cytoskeleton. We're going to walk through some regulatory mechanisms. We're only going to focus on the main lecture on microtubules and microfilaments. We're not going to talk a lot about intermediate filaments. Okay? So the cytoskeleton is a filamentous network of proteins that pervades the cytoplasm. It is everywhere in your cell. Okay? As you can see here, this is an EM of the cytoskeleton, and you can see it is this fibrillar mesh network, right? It includes both proteins that are what we would call self-assembling into a polymer structure, so monomers that will associate with one another through innate behaviors and form polymer structures. But the cytoskeleton is also considered, we also consider the proteins that bind to those polymer structures as part of the cytoskeleton. All right, so the protein filaments consist of these protein monomers that self-assemble. Again, it means that once those monomers come into contact with one another, they will associate and begin to polymerize. And so as you can imagine, the cell we have evolved mechanisms to basically regulate how well monomers come together. Because if we stick all these monomers in a solution with no regulation, they will form a polymer. It's their innate ability. So this is what we would term self-assembly into a final structure, and they disassemble to destroy that structure. Okay, so the assembly of filaments is regulated. They all grow or shrink by the addition or loss of monomers. Okay, this is what defines a polymer system. Monomers, everyone know what monomers are? Right? Anyone want to fathom a more formal definition? Yeah. So it doesn't have to be protein. So on this slide, I'm really talking about monomer versus polymer. They are going to be proteins in the case of cells, but yes. Yeah, so they're the building block, right? The smallest subunit with which will combine to form a polymer or strand. All right, growth is therefore referred to as polymerization. The extent of polymerization is regulated by three things in the cell. One is concentration. Obviously, the more monomer I have, the more polymer I'm going to have, right? Okay. If I have no monomer, I can't form a polymer, right? Those would be the two extremes. All right. We're going to talk about accessory proteins. In the case of microtubules and microfilaments, there are accessory proteins that are going to regulate both the activity, right? The activity of the monomers to self-assemble, and also the concentration and availability of those monomers. And finally, in intermediate filaments and in myosin filaments, which we're not going to discuss either today, there are events of phosphorylation that occur on those filament structures that regulate their polymerization. What type of enzyme induces phosphorylation? Kinase. You have two signaling guys teaching this class, so kinase is like the most important enzyme ever, right? Okay. All right, so what are the phases of polymerization? All right, so here we have our solution of monomers, and the first step is what we call nucleation. This is the rate-limiting step. Nucleation is when I go from a solution of all monomers to a nascent polymer being formed, right? The dimerization, the trimerization of 2 and 3 monomers, right? So we're starting to just build a polymer structure. And this is the rate-limiting step because these things are diffusing all over, right? And what we're requiring is that they're both active, they're both ready to form, to self-assemble, and they actually bump into each other, right? But if a polymer is going to grow from, let's say, head to tail, then they can't bump in like this, they can't bump in back to back, right? They have to bump into each other and interface with each other in the right way. And once that process gets started, once that nucleation process gets started, then we find the reaction kinetics sharply increase. So the next step is what we call elongation. And during elongation, this is a mostly linear growth process. I say mostly because I just mentioned we have a ton of accessory proteins that can jockey around with that rate based on availability and accessibility of the monomer. And finally, we reach this process of steady state, right? And so we talked a little bit about steady state when we talked about enzyme kinetics. It's the same principle, exact same principle here. So can anyone fathom a guess of how I would define steady state in a polymer system or in a polymerizing system? Yeah. Yeah, so steady state means that the association of additional monomers is equal to that of the dissociation of monomers, right? So that the fiber length, another way to state it, is that the fiber length should stay constant, right? You might be adding monomers, but you're also subtracting them at the same rate. This is what we would call steady state. So it's the same as the, remember we talked about Michaelis-Menten kinetics, E plus S to ES, right? And we made the assumption that that was at steady state, right? It's the same exact concept. We have a monomer plus a polymer going to polymer prime, right? That polymer with one additional monomer on it, right? But you're also losing monomers. The reverse reaction is also occurring, which is P prime is turning into P plus monomer at the same time, at the same rate, okay? All right. Okay. All right. Went too fast. All right, so what that establishes is this concept known as a critical concentration. So we just defined this. Polymerization is a chemical reaction where the reactants are a monomer M, here's our monomer, and a filament. This filament happens to have 7 monomers in it. And the product, in this case, is a filament that's simply 1 monomer longer, right? So now this polymer is 8 instead of 7. We can actually, we can characterize that polymerization process through this equation, that the change in length with time is equal to the on rate, right? So K is constant, it's a rate constant. Here it's called K on. I also introduced you to a concept of an association constant, or a Ka. That's the same thing. So this is the rate with which monomer is associating with that polymer, times the concentration of this monomer. Okay, so that's one term. And then we're going to subtract the rate with which that polymer is shedding monomers. Right? All cytoskeletal filaments are non-covalent associations between monomers. We're not covalently binding multiple monomers. You can imagine that would be much more difficult to disassemble, right? The K off would go not to infinity, but pretty high. All right. So we can define the critical concentration then as the monomer concentration at which the rate of monomer addition equals the rate of monomer loss. This is just what we talked about. The critical concentration is the point with which we have steady state. Does that make sense? So we're losing monomers as fast as we're gaining monomers on that polymer. Okay, so if we say that, right, that the polymer length is not changing, that means the dl dt, the change in length per time, equals 0. So we're going to do that here. It's equal to 0. It establishes this as a pretty simplistic equation. K on times this steady state concentration of monomer equals the K off. We can then solve for this concentration of monomer at steady state as being equal to the K off, the rate of dissociation of monomer from the polymer, divided by the rate of monomer addition to that polymer. So the K off over the K on. Or you might see this as the Kd, the dissociation constant, over the Ka, the association constant. Okay? This concentration of monomer is known as the critical concentration. Why this is important is if our concentration of monomer in this paradigm is larger than the critical concentration, then we're going to have a net addition of polymer. We're going to get polymerization. So when we jack up the availability of monomer, we start driving polymerization. If we drop the concentration of monomer below this critical concentration, then the fiber's always going to shrink, okay? Except that we have all these accessory proteins that regulate this kind of stuff. But from a basic polymer polymerization process, this, excuse me guys, will always occur. Any questions about that? Okay. All right, so let's talk about microtubules and microfilaments. Microtubules were addressed, or at least introduced to you guys in the pre-lecture. One thing to remember is that microtubules are enzymes. They're enzymes that we call GTPases. What a GTPase will do is it takes guanidine triphosphate and it catalyzes the hydrolysis of one of the phosphate groups to form GDP, or guanidine diphosphate. Okay? So these things are actively binding GTP and converting it into GDP. And that conversion, that acquisition of GDP and the conversion to GDP are critical when we start talking about how this polymer will form. All right, you guys were introduced to this idea that there's a plus end and a minus end. The minus end is associated with what? Where is that stabilized? The microtubule organizing center. Okay, so the MTOC, or microtubule organizing center, is at the negative end, the minus end, and it's stabilizing all your microtubules. Okay, so tubulin dimers are going to bind GTP. Once in the microtubule, the GTP is almost immediately hydrolyzed. Right? This is an enzyme. It's meant to turn GTP into GDP. What happens then is that because of that, we form this little thin cap known as a GTP cap on the plus end where the dimers are being added to the polymer. Okay? The residence time, so you can think about the growth is going in this direction. The residence time of a single GTP-bound tubulin, after a certain amount of time, that GDP tubulin turns into GDP because of the enzymatic activity of tubulin. Okay, so we have this little cap, there's my mouse again, that has GTP-bound tubulin. These orange or yellow, depends on, I guess, what you want to say. These are all GDP tubulin. So these are the tubulins that have already catalyzed that hydrolysis reaction. So new GTP-bound tubulin adds to the growing microtubule on the plus end, creating this GTP cap. The GTP cap, this is the critical part, the GTP cap has a much lower critical concentration than the rest of the microtubule. So we just talked about how I can jack around the concentration of monomer. What GTP tubulin is doing is it's lowering the critical concentration, right? So now at lower concentration of monomer, we have polymer addition. What that means is that if you don't continually add GTP tubulin to the plus end, and you give those molecules enough time to hydrolyze that GTP into GDP, now the critical concentration goes up, and it actually goes up above the concentration of the available monomer. And so it undergoes this disassembly. So microtubules are always undergoing growth, disassembly, growth, disassembly, growth, disassembly. They just don't stop. And so when the cap is lost, the microtubule is going to rapidly disassemble. And the reason is that there's not enough monomer here, right? A, we've consumed monomer by growing the polymer. And B, is that when we lose that GTP cap, we increase the critical concentration above what's available. So we get what's called catastrophic disassembly, or catastrophe. Okay? And during that catastrophe, if you're looking at it really carefully, you'll see they actually peel off from one another. They just sort of shed out, right? It's kind of interesting. It's like string cheese. Everybody likes string cheese? My kids love it, right? It just kind of peels apart. All right. Okay, so this establishes in microtubules what we call dynamic instability. So this refers to the process that a few rapidly disassembling microtubules feed the growth of many slow-growing microtubules. So here in this example, we have 4 microtubules. These guys, the green is denoting that GTP cap. So these guys, the Kc is low. This guy, he loses, let's say he stops growing He loses his GTP cap. He undergoes disassembly. Those tubulin will acquire a GTP, and now they can participate in polymerization of other fibers, other microtubules. Okay, let me make sure I'm... All right, so there's one thing that I want to make a note of here, that it's easy to think, because we're talking about the conversion of a guanidine triphosphate into guanidine diphosphate, and then the reacquisition of this GTP character. This is not a phosphorylation event. It's really easy to say, oh, there are 2 phosphates and then there become 3. You're just adding another phosphate. These GTPases do not function that way. So GTPases will always shed their GDP and are reloaded with a fresh GTP. And these are done by what we call nucleotide exchange factors. Okay? In the case of GDP, they're called GEFs, guanidine exchange factors. Okay? So they're going to completely get rid of the GDP, and another protein is going to facilitate the reloading of a new, fresh GTP. Okay? All right. So here's a video of dynamic instability. There's a kind of cool area up here. If you watch it, you'll see like one of these microtubules just disappear. So you can see they're growing slowly, and then they'll retract. Okay? And so the cell never stops doing that. Somewhere around here is the MTOC, the microtubule organizing center, right? So this is the negative end, and they're all growing here on the plus end. But then peeling back, yeah. No, it does not. It gets rid of, right? So it's going to first, so just for the recording, the question is how you go from GDP bound tubulin to GTP bound tubulin. So the GDP is going to completely dissociate, right? So GDP, tubulin has a lower affinity also for GDP than it does for GTP, okay? It's an enzyme. The substrate is GTP. It's going to bind GTP, right? So E plus S goes to ES, goes to E plus P. The reason that you get E plus P is that the enzyme has a much higher affinity for the substrate than it does the product, right? And so that product is released. GDP is the product of tubulin, okay? So when it makes that product, it gets released.